Thanks for joining, everyone. This is our monthly webinar for June. We'll be talking about the 8th Annual Invasive Species Mapping Challenge for New York State. And so the goals for today are to learn about how to participate in this challenge. So the two pieces of that are how to search for and identify the 6 focal species and how to report observations to iMap. And so we've got a lot of guest speakers with us today. We're going to have a really cool opportunity to hear from the experts on how to identify these species. So after my quick introduction, I'll hand it off to Maria Moscali from DEC, who will talk about beech leaf disease, which the DEC has been doing a lot of work on for the past few years. Then I'll pass it to Anna Dobson from Yale, who's been researching jumping worms. And then I'll pass it off to Ashley Morris, who worked for the DEC in Region 3, who will talk about some aquatic invasive plants. And then I'll give a quick iMap overview at the end, and then we'll have time for questions about the species as well as iMap troubleshooting if needed. So my name is Mitch O'Neill. I work with the New York Natural Heritage Program. We try to facilitate conservation of New York's biodiversity by providing information and expertise on rare species and natural ecosystems. We're a partnership between DEC and SUNY ESF, and we also manage the Invasive Species Database Program for New York State because invasives are such an important consideration for rare species and biodiversity conservation. And so me and my colleagues help administer iMapInvasives for New York State, which is our official database. And just to get everyone on the same page, invasive species are species of plants, animals, and insects, and pathogens that are, one, not native to the region, and two, have some sort of negative impact to the ecosystem, to human health, to the economy in the case of spotted lanternfly. And so iMapInvasives' role in that is serving as the centralized database for invasive species location information. So it's used by PRISMs, which are the partnerships for regional invasive species management, as well as state agencies, partners, the general public, volunteers, anyone working on invasive species issues in the state. And I just mentioned PRISMs, partnerships for regional invasive species management. These are really great. It's a really great network of organizations across the state, divided into 8 regions. If you haven't heard of your PRISM before, I encourage you to look on this map and figure out which one you're in, and then go connect with them, follow them on social media, that sort of stuff. They have a lot of great outreach programs, and they also work with partners within their regions to coordinate invasive species management. And so iMap is the official database for New York State. Data comes in a lot of different ways. In the past, a lot of it came in from partner organizations who had been collecting data since before iMap existed. But now one of our most important data sources is live data coming in real time from partner organizations. And also community scientists, like many of the people on this call. So we thank everyone who submits data to our database because that's what keeps the database up to date and accurate. And so what is the mapping challenge? So this is an annual community science challenge that was started in 2016. I think with one species back then, I think maybe just water chestnut. Then I think Tree of Heaven and Jumping Worm were added a few years after that, and then more species after that. But essentially, the goal is to fill data gaps in iMapInvasives. So we've been identifying species which are more widespread than the data suggests, so trying to get better data, better information on where those species are. And also to just grow our user group to better monitor invasive species in New York. So there are a lot of great organizations doing invasive species work in New York State, but New York State is enormous, and we really always need eyes on the ground from the general public to kind of help spread our coverage over the state of looking for invasive species. So community scientists are just critical to mapping invasive species. And so how the challenge works. Oh, it looks like I didn't update the dates, but we always start it on whenever we do this webinar. So it starts today, and it goes through the month of July. And so the species on our list this year are the same ones as last year: tree of heaven, jumping worm, beech leaf disease, nematode, and three aquatic plants: water chestnut, European frogbit, and watercress. And each year, depending on the data we get, a top prism will be selected to carry the trophy for the year. So you can see a lot of the trophies in this picture here. And also the individual winners get prizes too. So if you report the most of a certain species, you'll get a prize from the DEC. Here's some pictures of previous prizes. So um, issues with the conservationist, boot brushes, spotted lanternfly puzzle, that sort of stuff. So just as a thank you to our volunteers. And so where did these species come from? I won't go through each of the species right now because I'll be handing it off to our panel of experts for that. Essentially, these are all species where they are um, of interest to managers for some reason or another. So they have severe impacts, maybe, um, or little is known about them, and we want to monitor them. And some regions in the state do not have sufficient data for that species. So that's essentially how each of these species were added onto our list. So as an example, I'll just do a really quick um, uh, spiel on Tree of Heaven, since we won't focus on that for our ID portion of the webinar today. I'll just give a quick version. But you can see on this map, there are several regions of the state where Tree of Heaven is pretty widespread. The green is indicating municipalities where Tree of Heaven is present. And then we also have locations in yellow where people looked for Tree of Heaven but didn't find it. And you can see then there's also blank spaces in between those. So those are areas where we're not really sure. So maybe Tree of Heaven isn't there because it's not common in those regions, or maybe it's there and we just haven't looked for it or reported it yet. So we really appreciate the help to fill in these data gaps, particularly during invasive species challenges. And just a quick background on Tree of Heaven, since we won't have a section dedicated to it. Many of you may have heard of this because it's the bird host plant for the spotted lanternfly, which is an emerging species, um, uh, somewhat widespread in the New York City metro area and starting to spread beyond that as well. Been found in Western New York, Central New York, Long Island. But tree of heaven is also an invasive in its own right. It's native to Asia, and it's most common in disturbed areas, and it has some negative impacts like crowding out native species, and it actually uh, puts out pathic compounds in the soil that inhibit the growth of other species. And for identification, I'm just flying through it right now, but we do have more information on our challenge webpage, which I'll link to at the end of the presentation. But essentially, it kind of has this sort of palm-esque look to it with these leaves with a bunch of leaflets around them, pinnately compound. And one distinctive thing about each of these leaflets is that they have these little teeth at the base. I did a close up of that, yeah. So each of the leaves have these little teeth at the base of the leaf. and their bark is also very distinctive because it has a kind of cantaloupe pattern to it. Um, but again, that was a quick run through at our extended resources for better guidance on how to identify this species. But with that, I will go into our species ID component of the webinar. So first, I'll hand it off to Maria Moscalin, DEC, to talk about beech leaf disease.
So, American beech trees are an important component of the northern hardwood forest type, which makes up more than half of the forested land in New York, and wildlife such as black bears, squirrels, mice, turkeys, and many others rely on beech nuts as a main source of energy due to their high protein and fat content. And then predators such as owls and coyotes and others are also affected by the beech mast indirectly when their small mammal food source dwindles. Beech trees in New York have been in decline from beech bark disease since the 1960s, and now beech leaf disease is the latest threat. It can kill mature trees in 6 to 10 years and younger trees in just a few years. Loss of beech would cause changes in forest structure that would affect the whole ecosystem. Next. In North America, beech leaf disease was first found in Ohio in 2012, and it has been observed spreading outward from there since. In New York, beech leaf disease has been found on American beech, European beech, Oriental beech, and it's also been found on Chinese beech elsewhere. Next. In New York, DEC Forest Health first started hearing reports of striping on leaves and thinning in beech canopies in 2017. We started tracking it as beech leaf disease in the following year in 2018. At that time, the only detections were in the most southwestern part of the state, but each year since, we've seen a very significant expansion of the disease's range. So we have been so far tracking it according to county. And you can see in this map here, the counties. I added, we haven't updated the county map yet for this year, so I added a red dot in the counties that we found this year. And since I updated this map last week, we've, in addition, found it in Washington County. So we so far have 4 new counties that we found it in this year. We are also starting to map it out by the town level. So our interns and technicians that are surveying this year are trying to fill in the gaps on the town level. So as you guys are all out there looking, that's what would be really helpful, those areas to fill in. For example, downstate on Long Island, especially across Suffolk County, we know there's just crazy amounts of beech leaf disease like down there. So like, yeah, that's Suffolk. And then like really throughout Long Island, and then like especially Westchester, like Westchester is basically just saturated. So we don't really need as like reports from those areas. But any areas that are still open, that would be great to find out new places from. And when we wrap up our survey for the year, we'll put up some updated county map and town maps on on our DEC website as well. Okay, next. So since beech leaf disease has only recently been recognized, its biology and vectors are still not well understood. There's much unknown and research is ongoing. Research by Carta found that the foliar nematode Litolanchus crenati mccannii causes beech leaf disease symptoms, but it is unknown whether the nematode causes all of the damage or whether it associates with a pathogen such as a virus or a bacteria or fungus. So we don't know if it's just the nematode or if there's an association of different issues going on here. That's still being researched. The cause of its spread is also unknown. So we also don't know how it's spreading from one place to another. Birds or human spread are 2 theories. There has been research on birds, and they are coming up with some evidence. It's also just seeming like some of the places that we're finding it in are just completely in the middle of nowhere, where just like no one goes, like, like the technician is marking a boundary trail in the middle of a state forest, and is finding beech leaf disease, like that sort of thing. So it is seeming like bird spread might be like how it's happening, but still it's being researched, so we're not sure. Next. So now I'm going to go over what you need to know to survey. The ideal survey time is late May through October. So basically, whenever the leaves are out, they're going to be showing symptoms. The damage, there was research done and they showed that the damage happens inside of the bud before a leaf out. So right when the leaves come out, they already have the stripes. And that persists until they fall off, and then they even still have the stripes when they've fallen off. And so here you can see the banding at leaf out and again in the fall. So right now is a good time to be out surveying. Next. So this is the really important part, is that I need good, clear photos of the banding in order to confirm reports. If neither of these photos here, like I don't see enough evidence from either of these photos in order to confirm one way or another whether it has beech leaf disease or not. So I would have to mark this, either of these pictures, as insufficient to confirm. So anything without a picture, I can't really confirm unless it's like locally common. So if I get a report from like Westchester and it doesn't have a picture, then I put that it's locally common. Or like sometimes people put a lot of entries from one location, which please don't do that. But if you do, then sometimes I do locally common. But really, I just need 1 point per area. Maybe if you like were going down a trail and you wanted to do a point at the beginning of the trail and then again at the end of the trail because they're very far away from each other, that's fine. But I don't like, I mean, some people do submit like a lot of points and we don't need like all the different locations. Right now we're aiming for towns. So we're trying to confirm by like town level. But again, if you're in like a really large area, you could put in like one on one side and one on the other. So the really important part is that we want to take photos of the leaves as you're looking up through the light, that would show the banding. Next. I'll have examples of those. Maybe there will be pictures, I swear. Um, so this is identifying beech. The first thing you'll know is how to identify the beech, and beeches have that smooth gray bark. Unfortunately, a lot of them have had beech bark disease, and so you'll see like the bumpiness, or they're frequently carved into tree. Next. They're simple and ovate shape and have teeth, and you can see how every vein ends in a tooth, and the leaves are not hairy or sandpapery. Next. As compared to like Eastern hop hornbeam, you can see how each vein ends in a tooth, whereas Eastern hop hornbeam has like many more teeth. And like if it was elm, it would be like sandpapery and it would have those double teeth. So those are like 2 ways to tell beech. Next. So symptoms of beech leaf disease generally progress from the bottom of the canopy upward, but can be found randomly throughout. And likewise, it is usually found in the understory before the overstory. Early symptoms include the darkened striping between the leaf veins, and later heavy banding can cover most of the leaf surface. This will impact the tree's ability to photosynthesize. After a few years of being infested, leaves will become deformed, chlorotic, and have a thickened leathery texture. Next. So beech leaf disease symptoms are variable, so I'm going to show you several more examples of symptoms as well as other beech problems you might encounter. Here you can see examples of light to moderate banding, and the dark striping on leaves is most visible looking upward through the light. Yeah, so you can see these are the sort of photos that I want to see to be able to like easily confirm beech leaf disease. Those dark stripes. Okay, next. Here are a few more good clear photos of the dark banding. Forward. Next. Okay, so here, this is the slide I thought was going to be before. But why hold the leaves up to the light again? Here's a good example. Next. So these are the same leaves, but the first photo is taking a picture of the leaves looking down, and the second picture is taking a picture of the leaves looking up. And in the first photo, like I would think maybe that has beech leaf disease. It could, but I wouldn't know for sure. But then when I see the second photo, it's definitely clear. Okay, definitely beech leaf disease. So that's what I'm going to need to see in the photos that you submit. Next. Okay, so here, here's an example to spot an early infestation. See if you can spot it looking up into the tree. Next. Nice, yes. Next. Okay, here's another one. Try to find the banding. Next. Yep, next. Next. Yep. So there's like a few spots. So even if you like, I know sometimes you'll be like down at the ground, and you won't be able to like get right there to like take a close-up picture, but just do the best you can and like take it up through the canopy and like, I'll try to do the best I can to find the stripes in there. Next. So let's see. So heavily infested leaves are shrunken, curled, and leathery in texture. So this is like after it's been going on for like a few years. Next. And symptoms can progress to puckering, curling, chlorosis, and necrosis. Next. And then at the last stages, the leaves will be deformed, leathery, and possibly shrunken. Next. Okay, so now I'm just going to go over some other problems that you will probably encounter. These things are very common. They're not huge. They're not really like a huge issue for beech trees. They're just regular natural beech problems that our beech trees deal with regularly. So you will probably see them. So it's a good idea to like know what they look like so that you can tell the difference between that and the beech leaf disease when you encounter it. Next. And then again, let me just remind you that if you are unsure, it's better to submit a point with a really good photo if you're unsure than to not. Like if you think that it might be beech leaf disease and you're just not sure, just submit it. That's totally fine. Just make sure it has a good, clear photo. The first lookalike that might be confused with beech leaf disease is the Araneum patch caused by Areophid mites. And it's similar to beech leaf disease in that the damage is a gall, which is the tree's response to the organism. So that the gall forms on the leaf. This could be on the top of the leaf or underneath the leaf, but this is a different issue, and it's very common, and it's not that terrible for the tree. It's fine. Next. So here's just some photos like in comparison. I just tried to find ones that like make it that look similar. But for the Araneum patch, there'll be a felt-like area on the surface of the leaf, and there might be some puckering on the other side. And then most of the time I see light spotting like the top photos, but like when there's a lot of Araneum, there could be like the stripes. But beech leaf disease does not have that felt-like patch, and the thickened area usually takes up the entirety of the leaf veins, and it's a darker area and not a lighter area. Next. So next one is aphid damage. Again, this is really common. Aphids will cause a lighter banding or stippling between the veins versus the dark banding on the beech leaf disease. Next. So aphids also cause a curling of the leaf, but you can see in these comparison photos that the aphid curling has a much tighter curl along the side of the leaf, whereas the beech leaf disease curling is like kind of a deformity of the whole leaf curling. not just like the tip of it tightly curling. And then also, if you look at the underside of a leaf that has aphid damage, you might see some old aphid casings that they've left there. Next. The last lookalike for now is anthracnose, which on beech leaves looks like patches of necrotic tissue and usually on the end with the tip, but it could also be on the side. Next. And then here's anthracnose versus beech leaf disease. And then the beech leaf disease necrotic tissue could be along the sides or it could be presented as striping. Next. So treatments are being researched, but unfortunately nothing has been approved for usage yet. DEC Forest Health staff are cleaning their boots with a boot brush, and we are currently using Lysol. We used to use bleach, but now we just use Lysol on our shoes to prevent spread. This is out of an abundance of caution. We don't know that this actually helps. It's just that when we do our surveys, we go from beach stand to beach stand to beach stand all day, and so we don't want to be super spreaders. And if there's anything that we can do to possibly minimize that situation, then we're trying to do that. So that's what we do. We just get the soil out and spray the shoes with Lysol. Try to avoid moving the soil and leaf matter, and don't move firewood in general. Next. That's it. These are the references from the sources I've cited and the photos that I've taken. Next. For more information, please visit the DEC Beech Leaf Disease webpage. You can get to it from this link. Well, it's not actually a link, but this web address, or you can just, what I do is type into Google DECBLD, and it's the first thing that comes up. From there, you can also download, there's a beech leaf disease flyer that you can download if you want to give out outreach materials on your own, or you just want to like take a look at that. And then there's also the DEC Forest Health information line. If you have any questions, you can call that number or email or send an email to that email address. The best way, obviously, this is an IMAP presentation, but even if it wasn't, the very best way to report beech leaf disease is through IMAP. It's perfect. It keeps the data and location and the picture all together. It sends it right to the server, and it's really simple. So please just submit your points through IMAP. But if you have like additional questions about trees, you can call that number or email that email address. Next. Oh, and that's it. And let me just look at the chat. Okay, yes, so from Brittany Herman, I took a screenshot of the BLD survey maps for reference, but is there a website to look to see where the data gap towns are for beech leaf disease surveys? There will be. It will be the DEC BLD webpage. I mean, it's there now. We just haven't updated the maps recently. We'll do that as like soon, like because we're wrapping up our BLD survey with the interns, so we'll be updating those maps soon. Because the town map is from July of last year. We have more gaps filled in. Any other questions?
Yeah, and again, as people have questions, you can just go ahead and put them in the chat box. Thank you very much, Maria, for that presentation. Up next, we have Dr. Anise Dobson. Anise is a postdoctoral researcher with the Yale School of the Environment, and she's going to be talking a bit about jumping worm.
All right, thanks a lot. I'm really happy to be here, everyone. This mapping challenge is so useful to me in my research, both directly pulling data from IMAP, but also it lets us do these sort of big regional studies that would be impossible to do if we had to go and figure out from scratch where jumping worms are throughout the state. So I'm really grateful to all of you for participating in this, and And yeah, it takes the research a lot further when we have all of these citizen scientists in so many different parts of the state. All right, next. So a quick primer on jumping worms. They are earthworms, but they're different than the night crawlers, the more common earthworms that you're used to. This is in the family Megascelestidae, so it's a different family than the Lumbricidae that you're more familiar with. There are about 3 different quite common co-invading species in the state. Their taxonomy changes a lot, so the IMAP database people must be very annoyed with us because every couple of years we have a new recommendation for what it should be called. But generally they go by the genus Amenthus and sometimes Metafire. They're introduced from East Asia, and they're really different than beech leaf disease. So I'm glad that I'm coming in right after that great presentation. Beech leaf disease moves like this really fast wave. Jumping worms have been in the state since the 1940s, but have just been slowly building up their population and have seen a huge explosion over the last 10 or 20 years. Next. Their impact is really far reaching, and the reason for that is that they totally re-engineer the soil. So in a forest that doesn't have any earthworms at all, you'll see something like on the left, where the leaves from the deciduous trees fall down every year, decompose really slowly, and you have these really distinct horizons. It's a great habitat for salamanders and plants and fungi. You can see all these roots in there. And it has this sort of layered cake-like look. In forests that have those lumbricid night crawlers, it looks a little bit more like the one in the middle. These are also invasive earthworms, but they've been here a lot longer. And in those cases, it's going to look a lot more homogeneous as you move down the soil. And jumping worms, the soil looks entirely different. There's the very thick layer of castings in the top, 10 centimeters or so, that kind of look like these coffee grounds. They're really crumbly. It was hard to take a nice picture of the soil profile because the castings just tumble in. Next slide. So really all of the changes come from this re-engineering of the soil. This is quite gravelly soil for things to try and germinate in. You can see all these seedlings that have all these seeds that have tried to germinate but have dried out. So pretty destructive for the forest floor. Next slide. Some of the economic impacts, just really briefly, the castings can clog up drainage and stormwater drainage and ditches and things like that. You see big impacts to lawns and turf grass and golf courses and soccer fields, things like that. And then they can cause a lot of erosion with those castings being on the soil surface. Especially when you're talking about steep slopes or big rain events, you get lots of erosion. Next slide. And as might not surprise you, if you're re-engineering the soil that's at the bottom of the food web, it's going to have impacts that go to the plants, that go to the soil fauna, and then all of the things that rely on the soil fauna and the plants for habitat and food, etc. Next slide. Okay, so when you're looking for jumping worms, all year round, you'll be able to see these changes to the soil. So again, that uninvaded soil, sort of your classic historical New York soil, looks something like on the left. The European lumbricid earthworm, that's in the middle. And then the jumping worms with the very crumbly, gravel-like soil on the right. Next slide. And the reason that I'm starting with that is that all year round you can see that soil. The jumping worms themselves have an annual life cycle, and they do a lot of sort of expanding and retracting in times of like low rain and stuff like that. So you won't always be able to see the jumping worms themselves, but you can look for the soil. At this time of year, The worms are going to be really different in size. So we're over here somewhere close to the end of June, early July. Where I live in Connecticut, the jumping worms are approaching adults right now. They're pretty big. But here where I'm actually in Ithaca, New York, doing some field work right now, and they're very, very tiny. So depending on where you fall in this wheel, they're going to look a little bit different. And the other thing that you can see all year round is their cocoon. So that's where their eggs are, and it's about the size of a little pellet of fertilizer. Next slide. Okay, so first things first, you're looking for the granulated soil. Second thing to do is visually inspecting for jumping worms in late summer, early fall, especially after a rainy day, just under this year's leaf litter. So you don't have to dig down very far. You don't even really usually have to do any kind of a mustard pour or anything like that. Usually they'll just be right there. Next slide. And I'll actually ask you to press play on these two videos to look at the different behaviors. So it's a little bit, my internet's a little bit slow. I don't know if yours is any better, but the behavior is going to be the first sort of sign of the differences between the jumping worms and the lumbricid earthworms. So the jumping worms have this snake-like behavior. They really thrash about, especially when you first touch them. Sometimes they can drop their tail to avoid predators, but they have this kind of snake-like thrashing behavior. And then the lumbricid earthworms, they move along by expanding and contracting their setae as they go. And if they're disturbed, they're going to sort of slowly curl up and wind themselves around. So very different. Next slide. If you're a real taxonomy nerd and you want to start thinking about the differences in anatomy, then that can be really helpful to tell the difference. Right now, some of them will be adults depending on which part of the state you're in, and some of them won't be. But if you do find adults, you can know that jumping worms have this clitellum band very close to the nose of the worm, versus in the European worms, like on this one on the right, the lumbricid earthworms, it's going to be much closer to the middle of the body. They're both, the trouble is that they're both quite dark pigmented worms. They both can get to be around the same sizes right now. Sort of later in the summer, they can get up to about 20 centimeters, be about the sort of size of a pencil, even wider than that. So that's where finding that clitellum can be really helpful. And then sorry, I was a little too hasty on that. One more thing that I wanted to say is that the jumping worms are tubular all the way down, so they kind of look cylindrical all the way down, versus as the lumbricid earthworm, the European worm, moves, especially as it's propelling itself, it flattens its tail. Like in this picture, you can see the tail is quite flat, like a pancake or a beaver tail. So that can be a telltale sign that you don't have a jumping worm. Nice. All right, now we're ready. So just another side by side of some other different lumbricid worms and jumping worms. The shape of the clitellum can make a big difference. So again, that's this band here, the jumping worm close to the nose, the lumbricids closer to the middle of the worm. And then I'll get you to click twice here. Oh no, just once, just kidding. So the shape of the clitellum itself can be helpful. So in the jumping worm, it goes all the way around the body like a ring or like a corset flush with the body, versus on the lumbricid earthworm, it's just like a backpack, just on the back of the worm, kind of like a saddle. And then the belly of the worm, it's not exactly the same all the way around. We'll go to the next one. Now, at this time of year, if you're in places like Ithaca or further north, then they're really delayed this year, so they might not be mature, and so you might not be able to see that clitellum feature. But if you have a hand lens, you can look at the setae and these little hairs here on the worm that you can see at any life stage. In the lumbricid earthworm, they're usually paired or maybe close or widely paired, but there's usually around 8 to 10 of them as you go sort of around the circle of the worm. Versus in the jumping worm, there are all of these little hairs really stacked right beside each other in the ring around each segment. Next slide. Next slide. So as you're uploading them into IMAP, we have a lot of data across the state. It looks like it's pretty widespread, but unlike beech leaf disease, those local spreads really, well, maybe also for beech leaf disease, but the local spread really, really matters. Some places have had decades of jumping worms in one site, and then next door they don't have any. We don't totally know why that is. But taking multiple images is good. You can dispose the worms by freezing. That's kind of the most ethical, or heating, or putting in ethanol and then disposing. Then you can include additional observations about their behavior, etc., in the comments. Next. For photographs, if you can put a little bit of the casting in the picture, that really, really helps. The coffee ground gravelly type castings I mentioned. Mature specimens are great. Having good resolution, having an item for scale like a penny or a ruler is helpful. And then a contrasting background. The IMAP folks had a great idea of a white takeout container, which is really, really good. Straightening the worm, and then the more is not merrier in this case. So just 1 or 2 worms. 20 worms is not that helpful. They're harder to see. All right, I think that's the last slide. Oh, just quick summary. So when identifying them, first look for the soil, for that granular appearance and the thrashing behavior. That should be enough to get you where you need to go. At this time of year, if they're not mature, you can look for that bristle-like setae in the ring around each segment. If you have adults, then you can look for, in jumping worms, a ring-like clitellum, for kind of like the inner tube, close to the head of the body. All right, thank you so much, and I'll answer, for time, I'll answer any questions in the chat.
Great, thank you very much. So we'll just give one quick second to see if anybody has any questions. And if not, we'll go ahead and move on to our last guest presenter, Sarah Coney. Sarah is the Aquatic Invasive Species Manager from the Catskills Regional Invasive Species Partnership, and she's going to be presenting our 3 aquatic invasive species, the water chestnut, European frogbit, and watercress. Sarah?
Yeah, so I will be talking about these 3 species today. Somewhat common, becoming more common, unfortunately, here in New York. And we'll go over how to identify them and how to find them and report them on IMAP. Next, please. Yeah, so just like the terrestrial plants we've already talked about, aquatic invasive plants also outcompete native species, particularly competing for habitat and resources with a lot of our native floating plants, but they also form these really dense monoculture mats that can shade out submerged native plants as well. And these can have a number of effects on the physical attributes of our water bodies. So they can limit water flow, they can decrease dissolved oxygen concentrations in the water, change the temperature profile, and limit habitat for native animals. They can also, if you hit next, please. You can see from that, oops, I just wanted the water chestnut picture, sorry. Thank you. And so you can see from that bottom picture, that much plant biomass in one area can definitely impact recreation abilities on the water bodies they're in. Water chestnut and frogbit in particular, I have seen water bodies just absolutely choked up with water chestnut that it is impossible to kayak through, let alone put a motorized boat through. Swimming, not really ideal, and fishing lures will just bounce off of the water chestnut. Next, please. And so this is what water chestnut looks like up close. It grows in these rosettes. That's a very small one there on the right. They get about the size of a dinner plate. maybe a little bit bigger, and they will have multiple plants connected to one centralized mother plant, if you will. And they have these distinctive triangular leaves with tubed margins. People like to compare them to strawberry plant leaves. And even the flowers kind of look like a strawberry plant. You can see they have white flowers with 4 petals, a yellow center, and you'll typically see them blooming from June to July. After they bloom, the flowers are pollinated, they produce these nutlets you can see in that middle picture. When they're fresh, still attached to the plant, or when they first drop off, they're green and soft, but then they quickly harden and develop those really sharp spikes. And really important to note is these nutlets for Trapanatans, there are 4 spikes on each nutlet. So that's how they mostly travel around, are by these nutlets. They have very fine serrations on each spine that very easily catch up on feathers, waterfowl feathers. If you've ever been in the water with them, they'll catch up on clothes. Any clothing material, and they are very commonly found by especially the watercraft inspection stewards wedged between boats and their trailers. That's typically how they are moved around and introduced to new water bodies. Watercraft are a very significant vector for water chestnut in particular. Typically, you can find them, I find them in 6 feet or less of water, but they can grow pretty deep depending on the clarity and other conditions of the water. They are rooted to the bottom. And unfortunately, these are becoming somewhat more prevalent here in New York. They were actually introduced in New York in the mid-1800s first. And if you go next, please. And they've since spread around most of New York. However, there's a relatively new species called Trapa bispinosa that has been introduced in the last couple years. So this one, as the name is a really big hint to the key identification feature for bispinosa are the 2 spines on the nutlets you can see in that bottom left photo. Whereas again, Trapanatans, the one that is more commonly found in New York, and the only species of Trapa found currently in New York, has 4 spines on the nutlet. So if you are out surveying for water chestnut or you happen to find the nutlets, take a really good look and count however many spines you're seeing. If you are seeing the plants themselves, there are some physical features that differentiate it from Trapanatans as well. So you can see from those top 2 pictures that they have pink flowers with a yellow center, and the petals themselves are a bit more elongated compared to the Trapanatans. And they also have a nice reddish, purplish, pinkish underside to the leaves. So far, Trapa bispinosa has only been found in North Carolina and Virginia. But it is a very real possibility of it spreading. So it is something we really want to keep our eyes peeled for when we're out on the water. Luckily, there's nothing native that looks like any of the water chestnut species. Next, please. This is European frogbit. This is another invasive floating plant. This one has been causing me in particular quite a bit of a headache up in the upper Susquehanna. It was originally brought over for the aquarium trade and for water gardens. It's a cute little plant, has nice flowers, heart-shaped leaves. The leaves themselves are kind of thick. They're full of parenchymus tissue, air-filled tissue that helps them stay afloat. They also produce these little white flowers with, importantly, 3 petals, 3 white petals with a yellow center. And they are typically a free-floating plant. However, when growing in shallow water, they will root in the mud. And most commonly, we do see the roots tangled up with submerged aquatic plants like coontail or elodea. And while they prefer the surface and are most commonly found on the surface, usually mixed in with cattails, phragmites, or other emergent plants, you will occasionally see them growing a few inches below the surface of the water. When they're caught up with the submerged aquatic plants and there's rain and the water rises a few inches, they will stay submerged, and they seem to be happy growing submerged. So if you are out looking for frogbit, keep an eye in and amongst the cattails and other emergent plants where it usually is, but also be looking a few inches under the water as well, because you will commonly see the plants tangled up underneath the surface of the water. And so additionally, they send out stolons or runners with smaller plants, and those kind of tangle together to form these really dense mats, which makes them a bit easier to hand pull because you pull one plant and you usually get multiple. But again, that has the additional issue of shading out a lot of our native submerged aquatic plants. And interestingly enough, it's a dioecious plant, which means there are male and female plants. However, most populations in North America are either male or female. There's not a lot of sexual reproduction, so they don't usually spread by seed. They typically reproduce asexually, spreading through overwintering turions and fragmentation, those plantlets breaking off and spreading to new areas of the water body. We have been seeing them moving around again in the upper Susquehanna, primarily, we believe, through watercress. Those turions get caught up in the mud, get caught up in trailers. Kayaks and canoes are just as capable of transporting aquatic invasive species as motorized boats are. And so we have been seeing them move around in some of the wetland areas. They love shallow wetland water in particular. Next. And there are a number of native floating plants here in New York, some of them that do somewhat resemble European frogbit, especially when they're small. So two of the most common that you'll see out on the water are white water lily and yellow water lily, or spatterdock. So that top left picture is white water lily. The flowers are very distinctive. They are white with yellow centers, just like frogbit, but a lot more petals and different shapes. So those are a fantastic clue that you're looking at white water lily. The leaves themselves are much more rounded and have a very distinctive V-shaped cut with a pointed edge. And then on the top right is spatterdock, or yellow water lily. We have a couple different species native to New York. And they have a much more elongated leaf. When they are small, they do kind of superficially resemble European frogbit, the white water lilies as well. However, both plants get much larger than frogbit. Frogbit leaves get about the size of a silver dollar or smaller, whereas these can get larger than your hand, or about the same size as your hand. The yellow water lily also produces these pretty yellow flowers throughout much of the season. And both of these lily species are rooted to the bottom through pretty extensive rhizomes. Sometimes you'll see them floating on the surface of the water. They look kind of like logs almost, but they are very firmly rooted to the substrate. Another common floating aquatic plant is water shield. It gets its name. It has a very oval-like leaf, like a shield. And it has an absolutely fantastic coating of slime on the underside of the leaf and the petiole. If you've never seen water shield or felt it before, I highly recommend it. It's such a cool plant. It also has a pretty distinctive reddish, purplish underside to the leaf itself, and it produces these really pretty little pink flowers for a short time each season. And it's typically found in fairly somewhat acidic waters. And then lastly is little floating heart. This is the one that I think most closely resembles European frogbit. This is a native plant. It's not as common as the other three, but you do see it occasionally. I just saw a ton of it in one lake we were visiting that had a spectacular bog and the most little floating heart I've ever seen. And so you can, from the name, the leaves are heart-shaped. Again, somewhat superficially resembling frogbit. However, the leaves are typically thinner, less leathery, and more elongate. And the flowers, again, superficially resemble European frogbit. However, unlike European frogbit, which has 3 white petals, little floating heart has 5. It is also rooted to the bottom, unlike European frogbit, which is never truly really rooted to the bottom. There is an invasive species of floating heart called yellow floating heart, which again somewhat resembles frogbit, more closely resembles little floating heart, but it has yellow flowers. Next, please. And so the last species I'm going to talk about is watercress. This is probably one of the more commonly overlooked aquatic invasive plants, definitely here in New York. European frogbit stands out. That's one that we do lack some data for. So the more data we have for that is fantastic. Water chestnut is becoming common and is kind of a standout plant that it gets reported somewhat often. But watercress is very easily overlooked because it looks like any other dozens of herbaceous plants growing in and around wetland areas. Next, please. So it's in the Brassicaceae family. It's related to things like broccoli, cabbage, mustard, and just like those, it's also edible. So that's how it was believed to have originally been introduced, is escaping cultivation or discarded as food scraps. However, there are some nasty parasites associated with watercress, so I would avoid harvesting it from the wild if you ever see it. And so just like European frogbit and water chestnut, it came over from Eurasia. It's native to Eurasia, was brought to North America, and it has spread throughout most of the country, particularly along the eastern coast, but also over along the west. And it prefers shallow, wet areas, wetland areas, ponds, streams, particularly drainage ditches, puddles, and ephemeral areas. And so again, just like frogbit and water chestnut, it will form really dense mats that can crowd out a lot of our native species and can alter the physical attributes of our waterways. Typically, it is along the edges. At least when I am seeing it, is usually along the edges. It's not free floating out in the middle of the water body or out in the water like water chestnut and European frogbit. But in some wetland areas, swamps, bogs, and fens, it will be kind of growing in shallow areas in the water. And it will be attached to the bottom. Next, please. And so to identify watercress, it has an alternate leaf arrangement. So you can see in that picture on the right, the leaves are coming off alternate from the main stem, kind of. Typically has 3 to 9 leaflets with a wavy edge, and they're kind of rounded. And it produces these small white flowers with 4 petals, which is very common in that entire family, and slender fruit pods with the seeds arranged in 2 rows, which is another kind of important identification feature compared to a native species. So they don't have any hairs on the stems or leaves, and the roots are hollow. Next, please. So just like with European frogbit, there are a couple of native lookalikes to keep an eye out for. So the first one is the Pennsylvania bittercress. You can see the leaves are somewhat different, but the flowers and the seed pods are very similar. Next, please. And this is a state rare plant called lake watercress. So this one is not very commonly found. And this one, if you do find it, you should report it to New York Natural Heritage. But you can see it, again, has very similar flowers and seed pods, but the leaves are very different from nasturtium. Next, please. Watercress. So if you do have a watercress and you're not entirely sure what it is, you can check the seed pods. So the watercress seed pods are in 2 rows, whereas the 1 row, yellowcress, has just 1 row of seeds in the seed pods if you cut them open. Next. I think that's it. Yep. So yeah, watercress is very underreported, especially here in New York. You saw the map earlier. It's probably a lot more common than that implies. Same thing with European frogbit, especially in the Susquehanna where we've been working, in the Delaware, where I work in particular. It does seem to be very underreported. Water chestnut, that's one to always keep an eye out for because those nutlets can stay in the seed bank up to 10 to 12 years. So if you've been working in an area with water chestnut, you think you've eliminated it. If you're not continually checking to make sure it's not coming back, it can pop up and start a whole nother patch. So that's one to always keep an eye out for. And that's it. If you have any questions.
Great, thank you very much, Sarah. And special thanks again to all of our presenters for talking about our challenge species. So now that we've gone over all of the species, just to wrap up, we will do a quick overview of how to use iMapInvasives, how you can submit data. And then if anybody is brand new to IMAP, at the very end, we'll have a quick demonstration about how you can use the iMapInvasives mobile app to submit data while you're out in the field. So when you submit data to the iMapInvasives network, all of your reports will come in initially as unconfirmed. Basically what that means is you send a report to us that says, hey, I think I found this species out in the wild, along with a photo of it. And then once it goes into our network as unconfirmed, email alerts will be sent out to our network of confirmers. So the confirmers are individuals who work in the environmental field and are experts on certain invasive species. So things like beech leaf disease, if that gets reported, individuals like Maria will get notified. And then for ordinary species like tree of heaven, those are more broad. People like myself or the IMAP team will get notifications for those species. And once they are manually reviewed and the experts are able to verify that it is the species that you believe it to be, it will show up as a confirmed record and it will be available on the public map. So when you log on to iMapInvasives website, you'll be able to see that point right on the map view. And then from there, once it's confirmed, that will trigger email alerts if you have any specific jurisdictional email alerts set up for specific areas or for specific species. It'll show up on that map, like I had mentioned, and the data will then be used for our species tiers analysis. So for seeing if certain species are early detection or if they become more widespread, and allowing our partner organizations to strategize their management projects and prioritize their resources. So for reporting invasive species to IMAP, you want to make sure you understand which tools you want to use before you go out into the field. For most of our tools, they require a little bit of setup ahead of time, but then once you have them set up, you can use them out in areas that have limited service or no service at all. So the two main components is the online component where you will be setting up your account, and then the data collection tool component, which you're able to use on the mobile device and then use out in the field to report the actual species that you find. So the easiest tool to use is definitely the IMAP mobile app. So you can find that on the Google Play Store or App Store by looking up, New York iMapInvasives mobile app. Downloading the app using your iMapInvasives account information to log into the app. We'll go over how to make an account in just a little bit. Recording an invasive species using the Add Observation button, and then finally uploading your species to the database. And again, we'll do a really brief demonstration on exactly how to do this at the very end. So in order to use one of these data collection tools, you'll want to first set up an iMapInvasives account. So if you go to our website at nyimappinvasives.org, and then using the login button in the upper right corner, that will bring you to the login page. That will then prompt you to either use your existing iMapInvasives account if you already have one, or if you haven't logged into it in quite a while, you can reset your password. And then the larger box on the bottom of the page is for creating a new account. So all you need is a name, an email address, and a password. Your jurisdiction is where you're going to be collecting data. So for everyone here, it would more than likely be New York would be your jurisdiction. And then once you hit join, you should be getting an email that will have instructions to accept the user agreement. And then after you accept the agreement and then email, your account is active. And then you can use that account to log into our data collection apps and start submitting data to iMapInvasives. For one of our data collection tools, the Survey 123 form, it's a bit more complicated than the mobile app. The mobile app is definitely the most user-friendly and beginner-friendly of all our data collection tools and works for most scenarios. But if you want a more in-depth data collection tool, we have our Survey 123 form. In the follow-up email to this training, I'll send a link to a training for that as well, if anybody is interested in that. But for that tool, you will also need your account number. So once you make your account, or your person number, so once you make your account with iMapInvasives, you'll need to know that person number in order to submit data. So when you go to the iMapInvasives website, log in using your information, going to the menu, your account, and then that number will be there. And again, that is only for the Survey 123 form. The mobile app, you don't need to have that. You just need your username, which is your email, and the password for your IMAP account. Next. And again, if you want to download the mobile app, it's available for Apple or Android devices. It's also available for tablets as well. And like most of our presenters have mentioned throughout, good photos are definitely the most important part for collecting data. It needs to be clear enough that our confirmers are able to say without a doubt that it is the species that you believe it to be. So it's good to have a focus and close-up photo, also knowing those distinct characteristics that our presenters went over for each species, taking close-up photos of those characteristics. It's always better to have a close-up photo of a characteristic instead of a zoomed out and not too detailed picture of a population. And for most of our data collection tools, they will have a specific comment section where you can give more details about population size if you want to give that information as well. Next. And yeah, so that just about wraps it up. A few take-home messages. There are just a few invasive species to focus on for this challenge. Obviously, there are tons of invasive species out there, and you can report any of them that you see while you're out surveying for them. If you find any of the invasive species for this challenge, make sure you report them and have good, clear photos. You can also survey for these species specifically, and if you don't find them, you can submit not detected records for any of the species in the challenge, and that will also count towards the challenge. And again, the top reporters get a prize, and the prisms will receive a trophy for the amount of data that they submit. Any data that you collect and submit, either presence or not detected species, during the duration of the challenge, again, that's today until the last day of July, any data that you submit is automatically entered into the challenge. You don't have to mark it specifically or anything like that. Any data for those species automatically enters you into the challenge.
And this is Mitch O'Neill again. I just wanted to add one thing that Maria alluded to earlier that I forgot to include in any of my slides. But so we are encourage people to report, and the person with the most reports is who wins. But what we're looking for is reports at different locations. So like you're on a trail, and maybe you do one point at the beginning of the trail and then another point at the end or something. And then you go to another trail and report there. That's the kind of thing that we want to reward. We're not so much looking for like going to one place and creating, 50 points for each tree at that location or something. So yeah, just if you're going to a site and checking for species, you just kind of need to report that survey once. You don't need to report a bunch of different records. So I just wanted to mention that.
Great, thank you, Mitch. Yeah, that's important to note. In general, we try to keep it reports that are about 100 meters away from each other will be counted as individual reports. Obviously, if you have multiple populations that are completely separate from each other within 100 meters, you can report those both. They might just not both count towards the challenge. But in general, we try to count as much as we can within an area. So we also have a live dashboard up for the challenge at newyorkimapinvasives.org/challenge. Also, if you go to our homepage at nyimapinvasives.org, there'll be a banner at the top of the page that will bring you to the challenge dashboard. And as the challenge ramps up and people start submitting data, you'll start to see little charts and graphs pop up. Right now, it'll have no data. And the date range right now, it starts at July 1st. We're going to be changing that so it includes any data from today onward. But so if you collect data today or this weekend, it might not show up on that dashboard, but it is being counted towards the challenge. And within the next couple of days, it will be changed. So it will show up on that dashboard as well. But this will let you see the individual leaderboards for who submitted the most reports for certain species, as well as a aggregated data for each of the prisms. Yep, so on that gray bar there, you can swap between the two leaderboards. And that just about does it for our 9th annual invasive species mapping challenge. Thank you very much for attending, and thank you again for our guest presenters for going over our challenge species. At this time, if anybody has any questions about the challenge itself or any of the species, you can go ahead and put that in the chat. And if anybody is brand new to iMapInvasives and isn't familiar with any of our data collection tools, right after this, we'll be doing a very brief demonstration on how to use the mobile app that I mentioned earlier. So if anybody would like to stick around for that, you are welcome to. I'll go ahead and check the chat and see if there are any questions.
And another thank you to all of our guest speakers. Thank you so much for sharing your expertise on these species. And I know all of you have submitted reports to IMAP before, so you're all free once, if there's no questions, when Doug moves on to his demo, you guys are all free to go. Thank you so much.
All right, if we don't have any questions about any of the species, we will go ahead and move on to a quick demonstration on the mobile collection tool. So this is one that I had mentioned earlier. This is the most user-friendly tool that we have for collecting data, the iMap mobile app. You can find this in the Apple App Store or the Android Google Play Store. All you need for this is a iMapInvasives account, which you can make by going to our website. So once you make your account and download this app, it should automatically bring you to the preferences page where you can input your login information. Once you do that once, you are all set, and you can take it out to the field where there's no internet connectivity or no cell service even, and you can collect data points offline. So once you have that set up and you are ready to collect observations, you go ahead and tap the Add Observation button in the top right of the home screen. And if anybody has the app at home and wants to follow along, we're going to go ahead and do a fake species report, which you can go through the whole thing and you can submit the fake species report at the end as well. So once you hit Add Observation, the first thing that it will prompt you to do is add a photo. So you can either take a photo with your device if you are out doing a survey and you found one of the species of interest, you can take a photo of it. If you are doing a not detected record and like you've done a survey for the species and you haven't found any, you can just take a photo of the environment that you've been surveying in. You can also use photos that are already on your device. If you've taken a photo from an earlier hike, and then once you're at home, you want to submit a point, you can do that as well. And as long as you have location data on for your photos, it will automatically take the location from where the photo was taken and apply it to the observation. So you won't have to try and manually put where the observation was taken. Once you've selected a photo, there is a custom species list option in the preferences. So when you're setting up your app, you can set a custom species list. If you don't do that, it's just kind of a scrolling list where you have to, scroll through and find the species that you're trying to make a report for. But if you know you're just going to be surveying for these 6 species, you can go into the preferences and set a custom list ahead of time for just those 6 species. And then you only have to scroll through those 6 to find the one that you're looking for. For right now, if we're just doing a fake species for testing, that will be the first species on the list either way. But if you're going to be going out in the field, a custom list is good to have. And then So if you're doing it along at home, choosing a fake species, and then selecting whether it is a detected record or a not detected record. Again, so if you're out doing a survey and you don't find it, you can do not detected, but if you see it, take a photo of it and do a detection record. Those are the main points that you really need to fill out. If you fill out that information, the observation is considered complete enough to submit at that point, but there's a few extra things you can go through and check or add additional information for. So the location will be collected automatically, even if you don't have any internet service or cell service. It'll use the GPS location of your device to automatically put a point down for where you've collected your observation. And for the sake of submitting data on a mobile device, the location it gives you will be accurate enough. You don't have to worry about trying to make it precise or drag the little arrow to make it more specific. Whatever it gives you is good enough. And that will automatically fill in. Unless you don't have location services enabled for your device, in which case the coordinates it gives you will be 0, 0. And in that case, you go into your device settings and enable the iMap app to use GPS location. And then after that, there are a few more boxes at the bottom of the screen. If you scroll down from the map view, projects and organizations. If you are just a community scientist collecting data, you don't have to worry about these. But if you belong to any specific organizations or with any projects, you can put those in there. And those will be attached to your iMap account. Also, entering the approximate time that you were searching is a useful metric to have, especially if you're doing like a not detected survey. If you put that you were surveying a trail for 2 hours, then we know that the data is probably pretty good there, and we don't have to worry about doing additional surveys in an area. And then finally, there's observation comments. Like I mentioned earlier, if you want to give additional information of exactly how many of a certain plant you found, or what life stage they're in, if you saw some that were flowering and some that were producing seeds, all that information can go into the comments and it will be attached to your report at the end. And then the most important part is making sure you save your observation. Hitting save will make sure that it is saved onto your device. Since you're taking all this data offline, it'll save it onto your device for later upload. And when you're ready to upload your records that you took out in the field, once you're back home and you have Wi-Fi connectivity or good enough service to upload these, this is the process that you will go through. So if a species report is ready to upload, it'll show up as that little yellow box there. If you took a picture for it, it'll be on the left side of that yellow box. If there's any information that is missing, it'll show up as red, and then you'll want to go in and edit it to make sure that you fill in all the appropriate information. But once you're ready to go, your boxes are yellow and you're ready to upload. You either hit the edit button if you don't want to add anything, but if everything is good, there's a little checkbox below the edit button. You'll want to hit the checkbox there, and then go into the menu tab in the upper left. You'll want to hit upload selected, and then it will prompt you to make sure you are ready to upload. You'll hit OK, and then it will disappear. So if it looks like your record just sort of disappeared off your device, that's a good thing, so long as you hit upload selected. Once it's gone, then that means it was successfully submitted to our network. And like I mentioned earlier, it will show up as undetected, or sorry, not undetected, unconfirmed, until it is manually reviewed by one of our experts. And then after it's been reviewed, then it will show up on our map view. And that is the mobile app process. Fairly simple and easy to use. If at any point anyone has any questions about using the mobile app or collecting data, any of the species, anything like that, you're more than welcome to reach out to us at iMapInvasives at dec.ny.gov. Our email address is also on our website. You can reach out to us and ask any question. We typically respond within a couple of days. And yeah, that just about wraps up the training. I just want to give another shout out to our presenters. I think they've all logged off by now, but just want to thank them again. And thank you everybody who stuck around till the end. Thank you very much for attending, and we hope to hear from you soon and receive some data. And thank you very much and have a good day.